Okay, thanks very much, Alejandro. It gives me great pleasure to introduce Kris Wild. Kris came to us from the US in 2018 to do his PhD, and withstood a baptism by fire, being dumped out in Western Queensland for 2 years, chasing lizards. He came back, he survived all that, came back, and has been doing a really nice piece of work, supplementing his field ecology work with physiological work and more recently genetics. Kris represents quite a pedigree for UC and indeed the IAE. He's in fact the academic grandchild of Arthur Georges. Having studied with John Rowe at North Carolina University, and John Rowe was a PhD student of Arthur's in the mid-noughties. So Kris is nearing the end of his PhD time. It gives me a lot of pleasure to introduce him and to hear all about the work he's been doing in the last 3 or 4 years. Thanks, Kris.
Okay, cool. I just want to make sure everybody can see PowerPoint. I think we're good. Thank you all for tuning in virtually. I've had the awesome opportunity to work with this model species where my projects really piggybacked on previous work done at UC with the combination of laboratory and theoretical work conducted on this species. And it's allowed me to ask some interesting questions that not only relate to the species, but questions relating to the evolutionary transitions between sex-determining systems. So with every PhD comes with a long-winded title that tries to encompass all of your work that you've done over the past 3.5 years, or at least for me, I had a hard time figuring out, well, what do I name this thing? There's a lot going on. So really what I'm trying to get at is, I'm interested in if sex reversal is increasing in wild populations, and in particular, how individuals' behaviors and fitness of these individuals may play a part of the story. But before I get into the story of the dragons, I want to begin today by acknowledging the Ngunnawal people, the traditional custodians in the land that we meet today virtually. I pay my respects to elders past and present, and I extend my respect to Aboriginal and Torres Strait Islanders people that are here today. And second, I'd like to acknowledge the Kunya people, which are the traditional custodians of which the land much of my work was conducted during my PhD. Next, I'm going to spend a little bit of more time here than usual. I'd like to thank my supervisory panel. They helped me develop a lot of the work that I'll be presenting today. So thank you, Steve, Arthur, John, and Lisa. Other members or external members on my thesis has been Dan Noble. So he's really been helpful with some of the energetic questions that I'm asking with the species. So thank you. Also, the AWC, or the Australian Wildlife Conservancy, they really housed me and put me up in some really great digs while I was out in the bush over 18 months. So without them, the field component would have been a lot more difficult. So thank you. And then funding resources that allowed all of this work to be possible. It really allowed me to ask some interesting questions and use some really cool gear to get to those questions. And then finally, I'd like to take time to thank my family throughout my academic career. Today is a pretty big day for me. And then lastly, my family, so my partner Mallory and my furry friend Bonnie, that have really kept me grounded throughout my PhD. And then finally, this research wouldn't have been possible if it weren't for the extra hands that I had in the field over the past 3 years. So this ranged from international students coming to just go look at lizards in the outback to Australian students. So they really allowed, their work really allowed me to ask some really detailed questions and really relied on them to do a lot of the work. So thank you, everybody. And I'm really excited to share the story today. And really the story of sex reversal begins with understanding the different modes of sex determination. So sex in vertebrates is typically determined genetically by chromosomes. So in the case of humans, they exhibit male heterogamety. And all this means is that the difference between males and females is the presence of the Y chromosome. So the offspring of these two individuals will then inherit some chromosome, one chromosome from each of its parents. And there's another form of genetic sex determination where the difference between sex is with females. And this is where what we call as female heterogamety. So here we have the chromosomes that are ZW or ZZ for males. And the W chromosome is the difference between males and females. So the lizard that I'm going to be talking about today exhibits female heterogamety. So in a genetic sex determination system or a GSD system, it might be generally assumed that there's a 1-to-1 sex ratio and that they're resilient to sex skew. And on the other end of this, we have environmental sex determination, which is the establishment of sex by a non-genetic cue experienced in a discrete period after fertilization. So environmental factors which influence sex determination during development can include some sort of external factor like temperature. So in the example here, I have a nest of maybe salamanders or frogs and how temperature may alter the sex. So on the extremes, we get all males at cooler temperatures. And then on the other extreme, warmer temperatures, we get all females. And then kind of in the middle, we get a good mixture of each sex. And we can think of what's on this x-axis to vary. It's not just temperature, but it's the most common one. Variation in pH can alter sex ratios, nutrient availability, water availability, light intensity, and photo period. So vertebrates show an astonishing array of mechanisms that govern sexual phenotype. The boxes on this cladogram indicate modes of sex determination found in each taxa. So the colors represent if they're male heterogamety in blue or female heterogamety in pink, and if they occur within a species in that clade. Notice that species such as reptiles, amphibians, and fishes have species that contain both forms of genetic sex determination, whereas males and birds demonstrate strict male heterogamety or female heterogamety. So now I've added a box for environmental sex determination. The various environmental sex determination cues are indicated in red. Notice that groups like fishes have a wide variety of cues that can determine sex, where some groups have strict environmental sex determination like crocodilians that really are derived by temperature. So the most common form of environmental sex determination is temperature, and this is what we'll be discussing predominantly about today. Because sex determination is a fundamental process and is important for the development of individuals and the formulation of sex ratios in natural populations, taxa that contain all modes of sex determination provide excellent models for exploring the evolution of sex determining mechanisms. So this would include our fishes, amphibians, and reptiles. And rather than thinking about sex determination as a dichotomy between GSD or ESD, a continuum occurs from GSD to TSD, and that a critical influence can be genetic, environmental, or an interaction between the two. And the interaction between the two occur when sex is determined genetically at fertilization, but can be overridden at a crucial part of ontogeny by environmental factors like pH, water availability, or temperature. So in the case of the bearded dragon, temperature can override the genetic sex determination process. One major outcome of such a transition between sex determination systems is the potential elimination of sex chromosomes over time. So research has shown that this has occurred frequently among vertebrates and on an evolutionary timescale. So these are cues to environmental sex reversal, or where genetic sex determination can be overridden by environmental factors. So they include biotic cues, such as in the case of the clownfish, there's a structural change, a social change in the system where a dominant female maybe dies, and a male can undergo a complete sex reversal event and turn into a phenotypic female. The second being abiotic cues are the more common. So in the case where the bearded dragon, that's genetically determined. So here we have a graph on the bottom of our fishes of the proportion of males, and notice that it's 50% across the incubation temperature is about 29 degrees Celsius. We get ZW females and ZZ males. But when temperature overrides this process, it causes everything in that nest to be female, but half of those females will have the genotype that say that they're male. But if we were to look at these individuals out in the field, we wouldn't know that they're sex reversed or not. They would just be phenotypic females. So this phenomenon has been documented across various taxa, and new species are being added to the list each year. We're beginning to target species that experience this phenomenon, and we're beginning to understand the consequences of these transitions in nature. So we do know some information about the consequences of sex reversal, and they can be variable depending on the topic of interest. So for the case of morphology, or how the animal looks, in fishes, hormonal changes following sex reversal in the choho salmon have shown to provide rapid growth. Whereas on the other end of this, we get Nile tilapia that show a suppressing growth following these hormonal changes following sex reversal. And then we get things like frogs that experience no difference in body size once they undergo sex reversal. Depending on the species, there's variation in reproductive output as well. So in blue tilapia, sex reversed females have shown to have a decreased reproduction output following sex reversal in comparison to their female counterparts. And then with African pygmy mice, they're relatively interesting because sex reversed females have shown to have a increase in reproductive output in comparison to their female counterparts. And behavior of sex reversed individuals has shown to be different as well. So with those same African pygmy mice, researchers found that aggressive behaviors associated with sex reversed individuals allowed them to drive out competing females, or non-sex reversed females. So all of these cases take into account the individual response to sex reversal, and depending on how these sex reversed individuals compare to their phenotypic counterparts, this can shape what we see in natural populations. And we're beginning to understand how these individuals may shape population demography through a combination of approaches that include broad field studies and theoretical approaches. So we know the distribution of sex reversal can vary across geographic space, and this has the potential to alter population ratios. So the figure on the very top is a local lizard found outside of Canberra. And with this species, they're generally genetically determined, but cooler temperatures override this process and cause sex reversal. And what researchers in our group found is that the frequency of this sex reversal coincides with an elevational gradient. So the higher elevational populations have a higher rate of sex reversal. And then as we go down the mountain, we get less and less frequency of sex reversal. And to really kind of demonstrate what this can do to a population, recent work looked at a theoretical system that included Z males and ZW females, and how the proportion of sex reversal may drive a theoretical population. So the two panels here are looking at different proportions of individuals within a system that are sex reversed. So starting from the left, we look at the proportion on the y-axis and then generations on the x-axis. Each line fitted is the proportion of that sex. And in this case, the sex reversed individuals are in red. So the panel on the far left is looking at if there's 20% of individuals within a population that are sex reversed. What they found is systems stay relatively stable. But as soon as you shift that proportion from 20 to 50, it has the potential to rapidly drive down the W chromosome, or the ZW female in this case, and completely alter the population in this theoretical example. But in spite of the significance of this phenomenon, few studies have attempted to tease out the differences of sex reversed individuals and demonstrate how they relate to concordant male and female counterparts in the wild. So individual traits like reproduction, behavior, and morphology are important to understand because they may act as a motor or a brake for transitions between modes of sex determination. So the focal animal for my dissertation work has been the central bearded dragon. There's been quite a bit of work on this species in the laboratory and also these broader research questions on this species, but we know that the chromosome system is overridden by temperature in the wild. And recent work has identified a hotspot that's occurring in the southwest portion of its geographic range. So here's a map of where it's found within Australia. The colors indicate the sex. We're interested in the red ones. And work from our lab showed that the southwest corner had a higher than average frequency of sex reversal in compared of the rest of the distribution. And over the past decade in particular, this area has experienced an above average temperatures, and this species could be at risk of this phenomenon potentially increasing in the wild. So here we're looking at the past decade temperatures in Australia and how that relates across the past century. So what is really going on here is we need to understand how does this system typically work. So it's generally genetic sex determination where females are ZW and males are ZZ. I'll refer to these individuals as concordant sex because the phenotype matches the genotype. So these are, if we wanted to say the normal animals, but we'll call them concordant today. So in theory, under normal circumstances, population should be roughly 50-50, such as the nest above. So if these two individuals met in the field and did lizard things and the female nested, half of those individuals would be ZW female, and the other half would be ZZ male with the appropriate phenotypes and genotypes. However, if we took the same nest and incubated those eggs at warmer temperatures, we'd override the genetic sex determination process and cause conditions where we have sex reverse individuals. So these sex reverse individuals have the ZZ genotype, but they're phenotypic females. And depending on the conditions and the frequency of this process has the potential to decrease the W within a population. So to illustrate how we could lose a W within a generation, say we take the same nest that we had before, and we incubate these eggs above that threshold that overrides the normal genetic sex determination process. Notice now in this nest that we have all phenotypic females, but half have the chromosome complement that say that they're male. So that's indicated by the red ZZF. And say one of these sex reversed or ZZF survive to sexual maturity and happen to mate with another male, we get this example nest here, where we've completely lost the W chromosome within a generation, driving the offspring of this nest here to be completely temperature dependent. So mind you, this nest doesn't take into account what warm temperatures can do to sex ratios in this nest. So depending on the incubation temperature of this new nest, warm temperatures could create a scenario that could lead to all the offspring to be phenotypic female. So ultimately, with cooler or warmer temperatures, if this were to continue in nature, it could potentially skew sex ratios overall and decrease the frequency of the W chromosome within a population. So what do we know about this? So kind of starting with this theoretical models, we do know that if a population contains 50% of sex reverse individuals, it can quickly drive down the presence of the W chromosome in a matter of generations. What was shown here too in these theoretical approaches is that this phenomenon was exacerbated if there are differences in fecundity between sex. And when I say fecundity is if a sex reverse female could hold more eggs than a normal female. However, with these simulation studies, they identified that the immigration of the ZW genotype provided a majoring buffering effect for a transition to occur. So depending on how these animals are moving between populations may be an important mechanism to really halt that process. There are things documented in the lab that could potentially accelerate this process as well. So in the lab, we know sex reverse females have activity patterns and boldness metrics that are more male-like than concordant females. So here we have a series on the very bottom figure of behavior metrics of animals collected in the lab and the sex reverse females on the far right, and then our concordant sex on the left. And what researchers found is that sex reverse females showed bolder and higher activity patterns than their male counterparts for 2 of the 4 metrics. And we also know that body size of sex reverse females is larger like males, which could result to sex reverse females being more fecund and produce more offspring than concordant females. So this is almost like a positive feedback loop depending on what's going on in nature. And what we know in nature is this phenomenon is widespread across its range, and in particular in the southeast corner of its distribution, over an 8-year period, research showed that it increased by 16%. Finally, we know that isolation by distance is a primary pattern of genetic differentiation. So here, what this means is that as you increase in geographic space, the less related animals are. So on this plot here, we have an estimate for genetic distance and how that regresses across geographic distance. And notice this is a really large scale, but this is looking across its range. And what researchers found is that the species demonstrated isolation by distance. So depending on the dispersal potential of these animals, it could provide context if sex reversal can be spread through the movement of the ZZ genotypes, or if it's maintained by the immigration of the ZW genotype. So this leads me to what my questions were for my PhD. So based off of what's known from theoretical predictions, laboratory work, and temporal patterns, we need to understand these 3 questions in nature. So first, how do sex reverse females compare to their concordant counterparts, so the males and females, and fitness related traits? What is the frequency of sex reversal occurring within a population and across populations? And then how much migration and gene flow is occurring between those populations? These are 3 critical things that will inform the likelihood that environmental sex reversal is going to lead to complete chromosome loss in a system. So I've approached this kind of, or at least for the talk here, approached this in 3 different ways. First, looking at the individual response, then looking at individuals within a population. So this is just a figure that has the mixture of the 3 sexes that color coordinates. Blue is male, black is female, and red is sex reversed. And then how that may vary across populations. So first, going with the individual response, I was interested in the energetic requirements of hatchlings. And why energy? Because in some sort of fashion, animals use energy in every aspect of their lives. We can think of this beginning at the cellular level. So cellular respiration is a set of metabolic processes that take place in cells where organisms convert chemical energy from oxygen molecules into ATP, or really energy. Then an individual animal uses this converted energy from their food and assimilate it in a variety of ways, such as energy towards various behaviors, social interactions with conspecifics, and reproductive events. So as you can imagine, the uptake and the allocation of energy can vary across an organism's lifestyle. So for juveniles, they may invest more energy towards growth, whereas adults may, in this pie chart, shift this around and maybe allocate more energy towards reproduction. So we can then use these data to get an idea of the energetic constraints and how they may vary according to sex, which can provide context of how sex reversal is distributed across the landscape and provide potential mechanisms that act as maybe a motor or a brake for sex reversal, giving if there are energetic differences for sex reverse females over their concordant females. So first, we know that this is occurring in nature. We know that the morphology of these sex reverse individuals are more like males than females. So here I'll put a little chromosome that says that they're more like their genotype rather than their phenotype. As for behavior in the lab, we know that sex reverse females have similar behavior patterns to males. So again, they're more like their genotype rather than their phenotype. And then lastly, physiology. There's still a limited understanding of if physiology is affected by sex reversal within this species. And understanding physiology is, or particular energy use of hatchlings and how it varies according to sex will provide implications if sex reverse individuals have an energetic advantage over concordant females as soon as they hatch. These data are going to be used as a proxy if sex reverse females assimilate energy more efficiently than ZW female counterparts. Then this would mean that a sex reverse female may be able to grow quicker and allocate more energy towards various activities such as reproduction and acquiring mates. So our null hypothesis is if we look at metabolic rate on the y-axis and how that varies, or scales with mass, we expect that there's no difference. So the lines indicate what sex that we're looking at. So the sex reverse females being red, males being blue, and black being females. And then our like phenotype hypotheses, which suggests that sex reverse females are more like their phenotype rather than their genotype, and how how this regresses can be males can be more or less than females. And then the like genotype hypotheses where sex reverse females are more like their genotype rather than their phenotype. So this would mean that sex reverse females and males are more alike than concordant females. So we use stop flow respirometry to measure oxygen rates of hatchlings. We did this by taking air, scrubbing it free of CO2 with soda lime. This was then passed through a flow meter that controlled the total amount of oxygen within that chamber. So got kind of a picture of a lizard there, but the pictures on the far right are hatchlings in these really neat chambers. And then from here, we had an idea of the known amount of oxygen going into the system. So the animal was in that chamber consuming oxygen. Then we took the difference between the known amount that was in that chamber and then we pushed it through and allowed the animal to respire. And then we got these measurements of oxygen consumption per hour. So these measurements were taken overnight to ensure that animals were at a resting state. So if maybe there's some difference between activity that could affect their respiration during the day. So these were basal metabolic rates or resting metabolic rates. And the incubator that they were housed in maintained a stable temperature, so there's no effect on temperature and respiration. And then we used a Bayes approach to analyze these data and took into account individual as a repeated variable and accounted for day of samples as a random effect. So here we have the relationship between metabolic rate on the y-axis and mass on the x-axis. The color indicates sex, so black being concordant female, blue being concordant male, and red being our sex reverse females. The curves on top show the distribution of body size of our hatchlings. We found no difference across body size when we looked at sex. So, I mean, we measured these animals within 2 weeks, so we didn't expect there to be any difference. But overall, we found that there was no difference in metabolic rate across sex. However, when we account for mass-specific metabolic rate, we begin to pick up a pattern where sex reverse females and concordant females begin to scale more similarly as they get larger. So here are density plots that show the slope differences and how they vary across sex. The densities of the slopes are 95% credible intervals with means in the middle. Females and sex reverse females were shown to have a higher metabolic rate as they got larger. It would be interesting to follow up this experiment to see if this like phenotype relationship shows a stronger pattern as these individuals become adults. With sex reverse females being somewhat intermediate, again, it would be really neat to follow this up with adults and see if we find similar patterns or if they kind of maintain this intermediate relationship between the two. So when we looked at individual responses and we measured energetic requirements at hatchlings, we didn't really see a difference, but as they started getting larger, we started to see a scaling pattern across sex, where sex reverse females were more like their phenotypic counterparts. However, we didn't detect any kind of energetic advantage of sex reverse females over concordant females in respect to as soon as they hatched. So next, we were interested in looking at individuals within a population. So here, I studied a population for 18 months, and we looked at the frequency of sex reversal, and then we compared vital rates like body condition, survivorship, and how they varied across sex. And then we looked at movement. So here I was lucky enough to work at an AWC property called Bower Wildlife Sanctuary, so roughly 1,300 hectare reserve. So the image here demonstrates where it's at in relation to Australia. And it's actually on the edge of that hotspot that was identified this past year, with sex reversal being at a higher than expected rate within its range. So the site was selected because of that, and also it has a variety of habitat types. So this was also the first study on this species. So we really had no idea until 2 Americans go out in the field to figure out what kind of habitats the species was using. So generally speaking, if we look at the images on the top, Bower was this transition between sandalwood, shrubland, all the way to mulga, kind of tableland habitat. And this is indicated by the colors on the map here. We then conducted surveys on the property. So these surveys were conducted on using vehicles and on foot. So here we were interested in trying to find animals and using two different metrics. Vehicles, we recorded how far we drove and how many animals we found. And then our foot surveys were stratified, and we focused on each habitat type each week during the most active months of the lizards. So there's a picture of me trying to utilize my lizard searching abilities. They're pretty tough to find. But from there, once we found an animal, we determined their phenotype. So males have these pretty large bulges at the base of their tail. So they're hemipenes, whereas females have the absence of that. And then to determine if a female was sex reversed, we took a blood sample, and then this was sent back to University of Canberra, where we had some help, and gels were run to determine the chromosomal sequence. So here's a picture of what a gel is, and I've kind of figured out how to do this after my PhD. But the one dash line in that gel indicates that it's a male, whereas 2 indicates that that second bar is the W chromosome. So with females that were sex reversed, they would have the phenotype that said that they're female, but the genotype would, again, just indicate that they're male. We then fitted these animals with GPS backpacks, and this allowed us to track the animals out in the field and look at various behaviors and how they compared to these fitness-related traits that we're interested in, such as survivorship and movement. And then really compare how these data added up to laboratory and theoretical studies already conducted on this species. And this really provided us with an understanding of the conditions that may drive a rapid change from GSD to TSD. So first, looking at the frequency, I did mention these were relatively hard critters to find, but we did all right. So here's an example of me looking for an animal that actually had a transmitter on it. And given that they're relatively large animals, they're pretty cryptic. And if you haven't caught where the animal was at, he was staring right at me the whole time while I was trying to look for him. But we did all right. We captured 65 animals. Of those 65, 3 of which were sex reversed, 50 were males, so the concordant males, and then 12 were females. The map indicates the initial capture locations for each one of these animals, so they're distributed pretty evenly across the property. We had a few more individuals up front, but they utilized each habitat type. We then compared how this varied, finding these animals according to each survey method. So here we have a table that's looking at the 2 survey methods, so search, so looking on foot and vehicle, and then the total hours. So for both survey methods, we took over 3,000 hours to look for animals. And then lizards captured under that table, the bolded line, I'm sorry, the bolded number indicates how long it took to find one data point. So roughly 100 hours for either search survey or vehicle survey to find an animal. And then once we separated this by sex, we find that generally speaking, sex reversed females and females were found about evenly with both search and vehicle surveys, whereas males were a bit easier to find with our vehicle surveys because we were covering more ground in their behavior. But relatively tough to find females. They were like field gold in this case. So over 500 hours to find a concordant female and over 1,500 hours to find a sex reversed female. But from these data, what we can indicate that there's a male bias sex ratio. 20% of females that were captured were sex reversed. And then from there, we measured those animals to look at vital rates. So first we looked at how large these animals were and how that related to laboratory work, and then looked at if they were fecund, and what I mean by that is if they were gravid or not, and then looking at health indices such as body condition. So looking at this table here, we have 2 measurements of morphology, snout to vent length, which is just a length, and the mass of the animal, which is how heavy it is. Focus in, apologies for the table, but focus in on the 2 boxes, the red being sex reversed, the blue being our concordant male. And what we find is that sex reversed females showed similar body sizes and masses to males in comparison to female concordant females. So they were larger in size and larger in mass in comparison to concordant females. If we remember what laboratory work showed is that if a sex reverse female, well, sex reverse females were larger, therefore they were more fecund in laboratory studies. But what we find is that at no point did we find a sex reverse female to be gravid. And then when we looked at these indices for health, we looked at body condition. So it's just the residual difference between body size and body mass. And what we find is that females, although not significant, showed on average a poorer body condition indice than males. So with survivorship, we were interested in how survivorship varied across sex. I'd like to think of the bearded dragons as almost a common menu item for many of the native animals and non-native animals on the property. So it is a bird sanctuary. So we got really familiar with birds, but we also had events where other goannas, such as the sandy goanna, was chomping on a female. And then a few of our females that were nesting, we put nest cams in their area, and we found that cats were predated on them. And all of these photos were taken by myself or a volunteer. So it just indicates that they were predated on frequently on the property. So we looked at known fate models constructed in our mark. We were interested in a few parameters such as seasonality, sex, and body size, SCL. And then we looked at the combination of those and how they varied. Overall survivorship, females showed the lowest survivorship estimates, and sex reverse females were higher, but more similar to their phenotypic counterparts, and then males were at 78%. And then models that explain the most variation was the model that included the season-sex interaction. So this season-sex interaction showed the only support. And overall, with the exceptions of spring, sex reverse females and concordant females showed similar patterns of seasonal survivorship. So here, if we're looking at this graph, starting with spring, the lowest estimate are females, or concordant females. And then the open circles are sex reverse females. And then the line that kind of plateaus on the top are our males. Spring had the overall lowest survival probability across all sexes. And we find that this was really tied to their behaviors. And to look at their behaviors, we were interested in just daily movement patterns. So here's an example of 3 of our animals and how they moved on one part of the property and how that varied across days. And here we were just interested in daily movement rates. So how the movement varied between day 1 to day 2, day 2 to day 3. So we averaged that across individuals and then compared that across sex. So we looked at how seasonal movement and how that varied across sex classes. The color bars indicate the average daily movement for each sex across seasons. The connecting letters indicated if there is significance on a pairwise comparison for that season. So if they share a letter, they're not significantly different. If they don't share a letter, then they are. Based off laboratory work, we predicted that sex reverse females would have a higher activity or movement pattern than concordant females. However, we found the counter was true. Rather, the data indicated that there's no difference in movement patterns between sex reverse females during any season. And in fact, they were significantly lower than males in 2 of the 4 seasons. So these data indicate that there's strong differences in movement across sex, but they match the phenotype rather than the genotype. So with regards to individuals within a population, we got kind of a mixed bag here, but for the most part, our sex reverse females were more like their phenotype rather than their genotype. However, these data were limited because of the frequency of sex reversal. And come to find out, the genetic data actually provided potential reason why the frequency of sex reversal was relatively low. So here we're looking at the dispersal of these individuals across populations and also the genetic structure. So to do this, we used tissues collected within a 300-kilometer radius of our study site over a 16-year period to see how frequency of sex reversal varied across populations, genetic diversity, and dispersal potential varied across sex. So we used DART sequencing technologies to process these data. So here's a map of where Baura is that cluster of orange dots. and populations were assigned by the drainages that they were found in. So we do know that river drainages did provide a genetic barrier from a previous study. So we assigned populations based off of what drainage that they were found in between or around. The color indicates the population or dame, and the sample size is in the bottom right-hand corner. We first use a principal components analysis to visualize how populations assignments held up. And all we need to know with this figure here is if they were limited dispersers, you'd get a really nice clustering of colors on this plot. Whereas if they do disperse, you'd get some mixture of colors across populations. And what this PCA indicates is that there is some dispersal occurring across our assigned populations. So here we were interested on how dispersal may vary across our 4 assigned populations. So here we have the sample size of each sex, and the proportion of sex reverse females across the area was 15%. So first we used a more traditional metric that looked at FST values and how that varied across populations. And FST values are the proportion of total genetic variants contained in a subpopulation. So our subpopulation being the Peru, the Warrego, East or South. These values range between 0 and 1. And an FST value that's at 0 indicates no differentiation between our subpopulation, whereas a value at 1 indicates that there's complete differentiation. So an FST value that's at 0.01, we can think of there's 1 migrant per generation occurring in that subpopulation. So with this traditional metric, what it's doing, it's combining all of your data by your assigned populations, and we're getting a value. And we looked at that value and how that compared on a pairwise comparison basis across each drainage. So values that are above that 0.01, we can think of individuals that have moderate to limited dispersal ability, whereas values that are below that 0.01 means that there's high dispersal potential. So I'll save you some time looking through this table, but this table is looking at the pairwise comparison across sex. What it indicates is that no matter the sex, that there is high dispersal occurring across each sex. So the values that are below 0.01 indicate that the animals are moving. So now it would be interesting if we think about that PCA plot. Are the males moving across populations, or is it the females? So with the data that we have from DART, we're able to look at this at an individual-wise basis. So here, rather than assigning one population a value, we can look at how individuals may vary from the southern population to individuals that are maybe in more intermediate areas to individuals that are further up. So we did this by comparing the proportion of shared alleles and how that varied on an individual basis across sex. So the panels up on the right, white being our concordant females, our sex reverse females being in red, and then our males being in black. The p-values indicate if there's a significant regression between genetic distance and spatial distance on the x-axis. And what we find is that our females show that there's isolation by distance occurring on an individual-wise basis. Whereas males, that line is relatively flat, which indicates that they're pannyptic. So animals that are further away are just as related to ones that are found in the same area. So what this suggests is that dispersal is occurring within the species, but we weren't able to capture it with FST values. But when we look at it on an individual-wise basis, males are moving, whereas females are relatively sedentary in their dispersal abilities. So across populations, when we look at genetic structure, there's no difference in dispersal between sex reverse females and concordant females, which is important because if there was a difference where maybe sex reverse females were dispersing more, there could be an event that could rapidly cause sex reversal to happen across populations. So now I'm going to bring us back to the original questions of my dissertation. These 3 questions are really important to understand if sex reversal was going to completely transition systems rapidly within a system or across populations. So when we look at how sex reverse females compare to females and males and fitness related traits, we found no difference in hatchling energetics, sex-reverse females had similar morphologies to males, but really what we're interested in these indices for body condition and fecundity, and we found no advantage for sex-reverse females in comparison to concordant females. We found that, yes, survivorship was higher than females, but overall they showed the same patterns. We detected no difference in movement rates between females. Then we looked at the frequency of sex reversal occurring within a population and across populations. This is where it gets interesting. So within a population, we detected the proportion of females that were sex reversed were 20%, whereas across populations, the proportion of females that were sex reversed were 15. And really, we were building off of previous work that was conducted in 2011 that suggested that there's a potential that it could be increasing in the wild. However, our values, whether it's studying a population or across populations, don't support that it is increasing. But values are above the frequency detected across its range over a recent study that looked at museum specimens and the frequency of sex reversal over a 37-year period, which was 12%. And this frequency is probably not increasing based off of the low but sufficient dispersal abilities of the ZW genotype. And it suggests that that's maintaining stability of the ZW genotype within a population and preventing a rapid increase of sex reversal within populations. So kind of to tie this up in a bow, I hope what we need to keep in mind is that a transition between GSD and TSD is not a unidirectional process. My data show that the combination of the residual presence of the W chromosome across populations and the lack of evidence of fit disadvantage of sex reverse females enabling a transition between one that is genetic to one that is temperature rapidly. If either of these 2 points were true, we'd expect the system to be highly sensitive to such transitions. However, at least for some species that are GSD that have sex reversal, global warming may drive a transition to TSD. And the study highlights the difficulty of measuring such responses. So a species that lacks the dispersal ability would be at risk, or depending on how rapid warming occurs, some species may lack the time to evolve and optimize the thresholds needed to deal with sex reversal. And my work really highlights that the results of this study run contrary to several key expectations arriving from laboratory experiments and highlight the importance of measuring these processes in the wild and provide more, my data really provide a more robust comparison to test these predictions based off these theoretical approaches. So it would be really interesting to include these data from my study and apply them to the same models and see how this species now fits with this new information. And then areas that we need some more pieces to the puzzle is really understanding juvenile fitness and really nest temperatures in the wild. This would give us an idea of how frequent this is occurring and if early selection processes are acting on what we see in adults. Another avenue that I'm interested in is looking at the relatedness of sex-reversed females over time, which could give us an idea if all these individuals are coming from the same area or from one nest, and if they're contributing to genetic diversity within the population. Or are these females just, these sex-reversed females, more male-like, so males are avoiding them and they're just the function of an offspring that's surviving to be an adult. Then lastly, the energetics experiment should have a follow-up on adults to see if patterns hold up through sexual maturity. So do we get this like phenotype pattern with energetics, or do sex-reversed females kind of stay intermediate in between? And this could have implications on selection or how selection may act on sex-reversed individuals in the wild. And then lastly, it would be great to compare how these findings hold up to other species that have similar processes and really target ones that have different life history traits than our dragons. And this would give us an idea of the factors that may drive this process more generally in nature. So I'll leave you here with my key question slide from my PhD and the general results for each question. Overall, I feel laboratory and field data collected from my thesis provided a more robust comparison to test these predictions derived from previous work conducted on the species. This has allowed us to improve the understanding of the ecological roles sex-reversed individuals play on shaping population dynamics in nature. and overall has provided information on the evolutionary significance of these individuals under natural conditions. Thank you all for your attention today, and I would like to take some questions.
Thanks very much, Kris. That was great. Are there questions from the floor?
I actually have a question.
Cool.
Kris. So I wanted to pick your brain about the relationship between survivorship, fecundity, and nesting behavior, because you didn't find any ZZ females who were gravid, and you only found concordant gravid females. Did you find any evidence of increased predation due to nesting behavior?
That's a really good question. And I wish I had more time to really untangle these things. So yes, we found that survivorship was the lowest in the springtime, and this coincides with females doubling their movement at that same time. And that's when they're actually making these risky moves to nests or to nesting locations that are very much outside of their normal locations. And it's interesting because this could actually decrease the frequency of the W chromosome within a population if these females are really taking the risk to go to these pretty far nest locations. So the nesting locations seem to be really important, and there maybe is a trade-off there between survivorship and making these risky movements. And then sex reverse females were moving the same way at that time. So it begs the question of why are they moving if they're not gravid? But again, limited sample size on those behavior metrics. But again, we did see patterns, whether it's looking at the behaviors or the dispersal ability between females.
Great, thanks, Kris.
Cool.
Are there more questions? A lot of comments in the chat, Kris, but it's all praise.
So I want to, okay, I can pull up the chat. Okay. Are there any other questions? Smashed it or nobody cared?
No, there's nothing in the chat as well. Sorry.
Yeah.
I did say that your explanation of gels was great, and I very much enjoyed it.
It's important to understand that because going into the PhD, I did not really, I've run gels before, but I really didn't get why I was doing it. All right, well.
Okay, I think if that's it, well, let's thank Kris for his time, and we're keen to read your thesis as soon as it's done.
Yeah, so some important news, some of the data has been submitted or we're about to submit to molecular ecology. So hopefully we see some of the results soon.
Well done, congratulations.
Cool. Thank you everybody for showing up and paying attention or not.
Great job, Kris. Thanks, Kris, that was amazing.
Thank you everyone, have a good afternoon and a great weekend.